In our last set of videos, we learned all about DNA, so DNA's structure and its basic properties. And we also walked through the process of DNA replication in prokaryotes and eukaryotes, very similar processes. In this next series of lecture videos, we're going to now talk about the processes of transcription and translation. So first we're going to introduce the concept of the central dogma for gene expression, and then we'll move into other topics related to transcription and translation. So for these videos on gene expression, I'm going to divide this topic up into 4 separate videos. So the first part will be an overview about gene expression, so a little bit about the history of studying gene expression as well as some fundamental concepts. Then in part 2, we'll look at gene transcription and modification and all the molecular details involved in that process. In part 3, then we'll look at gene translation, again studying the molecular details of the process of translation. And then in part 4, we'll come back to thinking about DNA mutations, the types of mutations and the consequences, now that we have the tools to understand how that works. Okay, so the question that we're interested in right now is this question of how do we go from genotype, so a DNA sequence, to phenotype? And this is a question that biologists have been interested in for quite a long time. So we're going to start out by going back to the 1940s to a set of experiments conducted by a pair of scientists named Biol and Tatum. So what these guys did is they conducted a series of experiments with a type of bread mold called Neurospora crassa. So it's this red bread mold that's pictured in the illustration here. And what they knew was you could grow this bread mold on a substance called minimal medium. So that's a medium that contains an energy source and it contains nitrogen. And when you grow it on that medium, this mold can actually make all of its own amino acids. So it doesn't need any particular amino acids, it can manufacture all of its own. And Biol and Tatum also knew that it's possible to use X-rays to induce genetic mutations into an organism. So they learned this from a guy named Mueller who worked with Morgan, right? And so if you remember, Morgan was very much in the business of inducing mutations in fruit flies. So what they did is, with their experiments with this bread mold, they applied X-rays to the bread mold, and then they took spores from the X-rayed bread mold, and they transferred those spores to what's called an enriched medium. So this is a medium that contains all of the different amino acids. And then they took the mold that grows on this enriched medium, and then they transferred that to 2 different types of medium. So they kept some on the enriched medium, and they also put some onto a minimal medium. And they looked for situations where the mold could grow okay on the enriched medium, but it would not grow successfully on the minimal medium. So what that told them is that their mutation had caused a problem where suddenly this mold went from not requiring amino acids to actually requiring that the amino acids were present. So then the next question for them was, all right, well, if it requires an amino acid, which out of the amino acids is required? So then the next thing they would do is they would transfer some of these samples to a series of minimal media, and in each case they would add back just one of the amino acids rather than providing all of the amino acids as they did on the enriched medium. And then what they found was when they ran this experiment and they repeated this experiment with different types of mutants, in every case what would happen is when they tested out all these different amino acids, any particular mutant would only need one out of those amino acids, and then it would be able to grow successfully. So I've summarized that over here on the left part of this slide by saying, so most of the mutants that they created could grow on this minimal medium plus one supplied amino acid. So their conclusion from this was that the small DNA mutations that they caused with the X-rays must have caused a loss of enzyme function for the synthesis of a single type of amino acid. So it wasn't as though the mutations made it impossible for these molds to make all the amino acids. Instead, there must be very specific enzymes associated with the production of each specific amino acid. And a couple of biologists actually built on this idea later on and used this to figure out specific biosynthetic pathways for different substances in cells. And so, this brings us to an idea for the process of going from a genotype to a phenotype. So what Biol and Tatum discovered is now called the Central Dogma, which is the idea that one gene leads to the production of one type of protein. And so just to give you an example of this, if we think about albino organisms, so since squirrels are popular this year, here's a picture of an albino squirrel. And by the way, there's a cool society called the Albino Squirrel Preservation Society that's very popular on college campuses, in case you're curious. In any case, so albinos are not able to synthesize a pigment called melanin because of a mutation in DNA that codes for an enzyme in the melanin synthesis pathway. So the idea here is, so here's actually the process of producing melanin, going from an initial compound that gets transformed to other compounds, that gets further modified, and eventually what you wind up with is a form of melanin that produces pigmentation. And so each of these transformation steps is then catalyzed by a specific enzyme. So each of those enzymes then corresponds to a single gene in the pathway for making that end product. So an albino organism could have a mutation in any one of these enzymes that then impacts the final phenotype that gets produced. And in that case, if one of these mutations then made it no longer possible to go from one substance to the next, then you'd wind up with an albino. So that's how you could go from having organisms that have pigment to organisms that don't have pigment and get two different phenotypes. So building off of that initial work by Biol and Tatum, we now know that there are two distinct stages to the process of gene expression, and those two stages are referred to as the process of transcription and then the process of translation. And throughout this course, we're going to be doing a lot of comparisons between prokaryotes and eukaryotes, and we just talked about how the process of DNA replication is very similar for prokaryotes and eukaryotes. However, the locations where transcription and translation take place are pretty different for prokaryotes versus eukaryotes, although the overall process is generally the same. So the overall process in both cases is first to go from DNA to RNA. So that's the process of transcription, and then go from RNA to the production of protein. And in prokaryotes, the DNA is loose in the cytoplasm, and so all these processes happen in the same space. So prokaryotic organisms will immediately go from transcription to the process of translation, and all of that happens in the cytoplasm. And then in contrast, in eukaryotic cells, the process of transcription happens inside of the nucleus, since that's where the DNA is located. And there's actually an additional step to the process of transcription that's illustrated here, the step of RNA processing after RNA is produced. And then that RNA is exported out of the nucleus, and the process of translation takes place in the cytoplasm, either on ribosomes that are floating free in the cytoplasm or ribosomes that will dock onto the rough endoplasmic reticulum. So with all of this, there's kind of something of a problem. So we've got 4 different kinds of DNA nucleotides, right? We've got A, T, G, and C, just for shorthand. But we have 20 different kinds of amino acids. So the question is, how do we get one out of those 20 types of different amino acids from just 4 kinds of DNA nucleotides? I hope you can see that clearly a one-to-one relationship won't work, right? We're going to run out of nucleotides after we've specified 4 amino acids. We still have 16 more to go. So what about a 2-to-1 relationship? So then, there's 4 possibilities in the first position, and then there's 4 possibilities in the second position. So we wind up with 4 times 4 different possibilities, which means 16 possible combinations. But that's still not quite enough to get up to 20 amino acids. So what if we extend that idea further to a 3-to-1 relationship between nucleotides and amino acids? So if we do that, then there's 4 times 4 times 4, or 64 possible nucleotide combinations. So that's more than enough to code for 20 different amino acids. And this is indeed what we find. So after RNA is produced, so we go from the process of transcription, so going from DNA to the production of RNA. And in this case, this picture is also illustrating that what's transcribed is this upper strand that's referred to as being the template strand. So you'll notice that the transcribed mRNA has complementary bases to this template strand. And it has a very similar sequence to the other strand, the coding strand. So then to go from transcription to translation to a protein, then what we can do is we can read groups of 3 nucleotides from this RNA molecule and interpret those as coding for an individual amino acid. So each of these triplet groups of nucleotides is referred to as a codon. So that's a sequence that codes for a specific amino acid. Okay, so how do we actually set up this translation process? There's 64 different possible combinations of nucleotides, but only 20 amino acids. So biologists spent a lot of time figuring out how to translate this alphabet. And this chart summarizes what they figured out. And what this chart shows is, again, different codons. And then on the right-hand side is a shorthand abbreviation for the amino acid that code will specify. And you'll notice, for example, that in this little box here, there are 4 different codes that all code for the same amino acid. So there's some redundancy here. And you'll notice a similar pattern across this table, right? The same thing's true here for proline, or for leucine, or for threonine, or alanine, or valine. So, and in all of these cases, the difference between the codons that all code for the same amino acid happen at the 3rd position in that codon. And so this property is referred to as wobble, the fact that it doesn't matter what's in the 3rd position, you always get the same amino acid in a number of these cases. So that's how biologically we wound up going from 64 possible combinations to just 20 amino acids. There's a little bit of slop in this translation process. So it's useful to look at a practice messenger RNA strand to learn how to do this process of translation. So I just pulled up a little text file here that I want to practice translating this for. So here we've got the 5' end for this mRNA strand starts AUG, then UCU, GAA, UAA, and then we've reached the 3' end of this mRNA strand. So for translating this, we need to find AUG in this table. And the way this table is organized, the first base in each codon is on the left-hand side. So the codons that start with A are in this third row down. And then the second base in the codon is in each of these 4 columns. So if we're looking for AUG, we're going to go to the first column, third row, first column, and then the third base is organized within this table. And so AUG is listed here down at the bottom. And this AUG sequence codes for the amino acid methionine, abbreviated MET here. And we'll talk a little bit more about this start designation fairly soon. So our first amino acid here is going to be methionine. So then if we want to look up UCU, so U is the top row, C is the second column, and it doesn't matter what the third position is, all of these are going to code for serine, or SER. Then we have GAA, so we're going to go down to the bottom row for G, third column for A, and GAA codes for glutamine, GLU. Actually, that's a glutamate, but GLU. And then our last one, UAA, so U top row, A third column, A. This codes for what here says stop. And that's actually not an amino acid. That's actually a sequence that says stop translating this RNA. We've reached the end. So in this case, we have a protein that's extremely short. It's just 3 amino acids long, and it consists of methionine, serine, and glutamate. So that's, in a nutshell, how the process of translation works. So one really amazing thing is that this code, going from DNA to RNA to protein, this code is almost completely universal across organisms. So this language is a universal language. And what that means is that it's possible to transplant genes across species and get those genes expressed. So here's some really amazing examples of that. Here's a tobacco plant that's expressing a gene from a firefly, the gene luciferase, that makes fireflies glow. And then here's another example of a pig that's expressing another gene from a jellyfish that also makes the jellyfish glow, a green fluorescent protein. And so if we think a little bit more about this, the evolutionary significance of this is that this genetic language from DNA to RNA to protein is a language that must have come about very early on in the history of living things. And so it's part of how all living things are connected, kind of coming back to that biology theme of unity. Okay, so that takes us through our overview, and in our next video we will dive into the processes of transcription in greater detail.